Hey, welcome everyone to our RootSnap webinar. Before we start today, just to go over a few housekeeping things. Our moderator today will be posting links in the chats as I talk about places to find more information or information to receive quotes or pricing. If you have any technical difficulties or there's something wrong with the presentation, please put those in the chat box. But if you have questions about the material or the presentation, please put that in the Q&A section. So the Q&A tool is right next to the chat on the screen. Please do not put any questions in the chat box. I will not be able to see them. I will only be able to see them if you put them in the Q&A section. So like I said, welcome to our RootSnap webinar. My name is Kendra Ellertson. I'm currently an application scientist here at CID Bio-Science. I have my bachelor's in biology from Chapman University, where I worked in a plant physiology lab for 3 years, and I conducted research on how the quantity and quality of light affects tomato plant growth. I also have a bit of experience in the indoor vertical farming industry, and I've been with CID Bio-Science for over a year. So let's get right into the importance of studying roots. So first of all, the importance of studying roots, it really gives us a holistic view of plant health. Half of the biomass of a plant is underground, so we can start to understand how this below ground biomass interacts with the environment and actually contributes to the growth and productivity of the entire plant. By looking at root systems, we can also track changes in nutrient uptake. Since root systems are the main pathways for water and nutrient uptake, we can really start to understand how plants are absorbing nutrients from the soil and interacting with that below ground environment. We also have the possibility to detect early stress and disease. So instead of only being able to see that above ground biomass and track those changes, roots can actually show signs of stress before shoots and leaves can. And so growers and farmers can get on finding solutions for those stress and disease issues if they're able to look at the root systems. And also, root health is related to overall yield. So a better root system is going to be directly related to biomass production and better quality of your plants. So at CID Bio-Science, we currently have a line of root and soil imagers. We first have our CI600, which is our in situ root and soil imager. And this device is supposed to be in ground. So we have clear plastic tubes that get installed below ground or in a planter or pot in your laboratory. And then this device slides right into that clear acrylic tube. So these devices light up, they illuminate along the entire length of the device, rotate 360 degrees, and give users a visible image of their root systems. Our CI600 can go up to 600 dpi in image resolution, and I've included some pictures over on the left here to give you an idea of the different soil types and root systems that these devices can measure. We also have our CI602. This is our narrow gauge root and soil imager. These devices are very similar, pretty much the exact same thing, except the CI602 has a bit smaller diameter, and it also can go up to 1200 dpi in image resolution. Both of these devices are meant to be placed in the clear acrylic tubes below ground, or like I said, in pots or planters in your lab. Both rotate 360 degrees and give you that visible light image. Along with our CI600 and our CI602, we are introducing our CI610 autonomous root and soil imager. So this device has not yet been released. We're still in, the phase of, figuring out how all the components are going to fit together, making sure we can give users a really good image quality and fit all of the tools and features that we want into this device. But just to give you guys a little bit of a summary and an introduction to this new release, this CI610 is going to be completely non-destructive, super portable device for below ground imaging. It's also going to utilize UV and NIR wavelengths as well. So unlike our CI600 and CI602, which are only using visible light, this device has the capability to use UV and NIR as well. It will also be completely wireless. So with the 600 and the 602, it does require a cable to be connected to your laptop or tablet for operation. But with the CI610, it will be completely remote operation that you can do from your mobile device, where you can tell the device when to start traveling down the tube, what images to take at what intervals. Also, you'll be able to send and tag all your images right on your phone. It's also going to possess the capability to do video capture as well as image processing. So when you start the device from your phone, the device will illuminate at the bottom and it will travel autonomously down the tube. And as it's traveling down the tube, it'll be taking a video. It'll detect that it's at the bottom of the tube and reverse and start to go back up the tube. And that's when it's going to do its image processing. And then once it reaches the top, you'll see those images and video on your phone. Check the links in the chat box for early bird pricing info to be up to date on when we do release this product. And we also are looking for beta testers. So when we do release this device, be sure to check out all those resources on our website. I wanted to talk about a few real world examples of people who are actually using our root and soil imagers. So just quickly, we have Dr. Lorenzo Rossi, who is at University of Florida, and he's using our CI600 to determine optimal nutrient concentration for citrus under HLB endemic conditions. We also have Dr. Elmi Lotza, who's at Stellenbosch University, and she was looking at how plant nutrition and climate change affect root dynamics across many species. We also have Dr. Rao, who is at Agricultural Victoria Research, and she's examining how pulse root system architecture, she's examining differences in pulse root system architecture for trait selection specifically. So with all of these users, once they've taken their images with the CI600 or 602, they need to analyze those images. Having the visible images is one thing, but actually being able to get data from those images and determine the density, mass of your root systems is really important. Luckily, we have a software to do just that. So our RootSnap software, just a little bit of an introduction. It's on our website for download. So if you want to follow along in the next part with me, you can go ahead and download that from our website. But with the RootSnap software, you can upload any image into this software. And by tracing out your root systems, you can get measurements of root length, area, volume, diameter, branching angle, etc. You'll also be able to see average values for specific tubes, specific root systems, or singular root systems. It's a very intuitive and easy to use interface. We've made sure that all of the tools and buttons on the software are, very clear and easy to use. Some cool features about this software is we have a time series analysis feature. So for users who are coming back different weeks, different months, different growing seasons to analyze and track changes in their root systems, tracing out these root systems takes quite a bit of time. And so we've added a software that if you take an image in week 1 and trace out all of those root systems, you'll be able to overlay those roots that you've already drawn out onto your next picture. So you don't have to go back and trace out those roots that you've already done. You can just add on to your project. We are in the process of making this software fully automated. So like I said, right now, the user does have to trace out all of their root systems, but we're in partnership with an AI company to hopefully make this fully automated. So, it will trace out all of your root systems and do all of the analysis for you, which would really cut down on time. So now we're actually going to go into a live training of the RootSnap software. I'm going to switch my screen to our RootSnap program, and I'll walk you through all the tools and features of that software. So here is our RootSnap software. This is what it looks like right when you open the software. You kind of have your blank canvas, your blank project to start. First thing I always recommend doing is going ahead and saving your project. Saving that into whatever file or folder system you'll be able to easily find it later. With this software, you can add multiple tubes. So let's say you've started a project for your apple orchard. You can add all of your tubes that are in that apple orchard right here. So you don't have to make multiple projects for multiple tubes. If you want to add a new tube, you can just click this plus button, and this dropdown will show you all of the tubes that you have, and you can easily delete them with this button here. We also have this sessions tool here. So this is great, like I said, for that time series analysis. If you want to track the progress in your root systems over time, you can add multiple sessions. And I'll be showing you a little bit about this later. I'm going to show you an example of what like a time series analysis would look like and how to overlay those root systems. Down here in this Windows section with all these boxes is where you can start importing your pictures. So for each tube and session, you're only going to want to add the pictures from a single tube. So I'm going to go ahead and just click into this box, and it's going to pull up my file explorer here. And I'm just going to add in some pictures. To move the picture around and zoom in and out, I am using the pan and zoom tool, and I'm using the cursor on my mouse to zoom in and out of the picture, as well as move the picture around. Before I start tracing out any roots, I want to make sure that I can easily see my roots and there's a good contrast between soil and roots. So I'm going to use these tools up here to adjust the brightness and contrast of my image before I even start tracing out anything. So you can use this toggle at the bottom to adjust the brightness. This middle section here is what your actual picture will look like, but we've added some examples on the side of some different levels of brightness that you can easily just click and it will apply. So I'll show you here. If you wanted to click this one, just click it. It will apply that to the main picture, and then you click apply. And if you have multiple pictures in your windows down here, you're going to want to adjust the brightness and contrast for all of them. Again, just to make sure you have a very clear contrast between that soil and root system. And I'm going to do the same thing with my contrast. Just make sure I can see them very clearly. So to start actually adding and drawing out root systems, you're going to want to zoom in to the first section on your picture. So I'm going to zoom in up here and make sure I have a good view of where I want to start. And then I'm going to select this Add Edit Points tool. So with any of these tools up here, a single click will select the tool, but then another click will actually give you some more options for that tool. So once I've selected this Add Edit Points tool, I'm going to click again to see these other options. This range tool up here allows me to adjust the size of my cursor. I always suggest starting with a smaller to mid-range size just to start so that you have a little bit better control of where you're placing your roots. I'm going to start with this smaller size down here. I also suggest that you have this snap to root feature enabled. So what this does is as you're drawing out root systems, it's going to detect color changes between roots and soil and snap your points to the middle of the root. Makes it easier for all your points to be placed on the root, and so you don't have to go back and manually edit each individual point. Now to start drawing out your roots, you're going to start at one end of your root and just click and hold with that tool and trace along your root system. And you'll see those points kind of snap into the middle of the root there. And I'm just going to trace out a few more here. And then we'll get into the specifics about how to adjust the points. So when you're drawing out these root systems, you also have the option to connect branches to your parent root. So it doesn't look like this root is connected yet that we can see, but I'm going to show you how to connect that as if it were a branch. So this last point here, I'm going to select it. And as you can see, I can kind of move it around wherever I want. If I drag it over another point and let go, it will snap those roots together. And now, as you can see, that is one connected root system. I'm going to zoom in and show you a little bit more about what these points are. So when I zoom in, it's going to give you a millimeter diameter. I can adjust these points, and you want to be very accurate here because this is where the software is going to actually calculate the data from these points. So you want to make sure these points are adjusted properly. You can adjust them in two ways. The first way is to just click on one and use the cursor on your mouse to adjust the size. So I'm just scrolling up and down to adjust that size. You can also click on it and move it around if you need to. So that's one way to adjust the size. Another way, if you click the View button up here, select Panels and Point Details, this panel on the right will show up here, which shows you the diameter of all the points that you have. Now you can actually select specific points and edit the diameter here. So if I wanted it to be 4, it will auto adjust that size on the picture and in the selected points bar over here. I can also shift and select multiple points and choose whether I want the software to estimate that or set it to a specific diameter, or I can even select my own. And it will change all of those. If I make a mistake and I need to delete, there's two ways I can do that. So I can either right click with my mouse and this red cursor will show up. And if I drag it right over my roots, it's going to delete those. I can also actually click on this tool. And again, I have some more options to change the range. So the size of my cursor here, I'm going to choose the mid size. As you can see, I can just delete. You also have some more options. So to split roots, this is a nice feature when this is enabled. If I hover and delete over where I've connected to roots and delete that point that's connecting, it's going to split those two roots back into separate ones. I can also choose to remove the entire root. Just like that, I've removed all of my roots. I'm going to draw a few more on here to show you some of the other features. I'm not being very precise right now, but when you do this with your own pictures, you're going to want to make sure that you're zoomed into the picture so you can very clearly see the diameter and length of these root systems, and making sure that your points are directly on the roots. So some other features, this lock points tool is really great if you have a lot of roots that you're trying to draw out on a picture. You can actually lock up your points so you don't accidentally move them or delete them while you're working on other sections of your picture. So when you select this, you're going to have this blue cursor. Again, you can change the size of your cursor. I'm going to zoom in to show you this a little bit better. When this point is selected and I go right over those points, as you can see, there's a little lock that shows up in the middle of those points. Now that means that even if I tried to delete them, I can't. They won't be deleted, but this one up here, since it's not locked, can be deleted. To remove that lock, you can just select, go over those points again with the lock tool, and it will unlock them. So now you can adjust and move them. You can also adjust the color scheme of your roots. So if you select one of your root systems and come up here to root color, we can change the color of any of those that we want. Another feature is this measure manually tool. So I like this tool for users who, don't want to use their mouse to adjust the point size of all their points. They can measure the average length of their system, their different roots, and then manually add that diameter to all those points here. But this is a nice, quick tool to look at width, length, just an added feature. So another feature of this software is if you want to kind of revert back to your original picture and see what it looked like before you started drawing out all of these colorful roots, this show hide roots tool up here, if you click it, it'll take off everything that you've drawn out so far. And if you select it again, it'll put it right back on. So once again, I'm just going to draw out a few more of these systems so that you can actually see what the statistics will look like at the end of the project. And again, to connect two roots together, you'll just select one of those points and hover it over another one, and it'll connect those together. Another feature that we have is this auto detect feature. So this is really great for high contrast roots that stand out. So with that feature, you're going to click the Add Edit Points tool and just draw a few points at the start of your root. Then you'll select this Auto Detect feature, and it's going to detect the contrast between soil and roots. And as you can see, it just drew out that entire root for me. So to start seeing some of the statistics of drawing out all of these roots, you can hit this View button up here, click Panels, and click Root Details. And so this is where you're going to see some of those specs for individual roots. So up here, this is going to show you all of the roots that you've drawn out so far. They're color-coded and numbered. Every time you draw a connecting root or, drag and drop one of the roots over your parent root, it's going to increment those in decimals. So the parent root will be a whole number, like it is here, 16. And then we have our connected root, 16.1. If I were to add another one, it would say 16.2. Like I said, these are all color-coded that correspond with what's on your image. And you can select any of these roots, and the statistics for that will show up on the side here. You can also change the status of that root. So if it's a new root, alive, or dead, you have the option to select any of those here. And as I toggle through different roots, obviously these specs are going to change. This branch angle section here, it will be 0 if you're selecting a single root. Once you select a connected root, that's when that branch angle spec will show up. To view the average of the statistics for your picture, again, you can click View, Panels, and click this Image Details button. And so this is going to be an average of this entire picture. It'll give you the total root area, and the average diameter. And then if you need to close any of these windows, you can just select this arrow button and those windows will pop back to the side. So now I want to show you how to do a time series analysis. So we're going to pretend that this is my first image that I've taken with my root imager. There's only a few roots here that need to be, drawn out, and I'll quickly draw out just a few more of these here. Then we can make a new session over in this toolbox over here by clicking the plus button. That will give us a new session. And here I'm going to add what would be our, let's say, week 2 image. So again, I just clicked on that gray box and I'm going to click our week 2 image and it'll pop up on the screen here. So like I said, I have already, drawn out some of these root systems in our previous picture, our week 1 picture. And I don't want to have to go back and retrace out the roots that I've already traced. So what you can do is you can come over here to this Windows section, right click on the picture, and you have this option to migrate roots. So if I select this, it's going to ask me if I've drawn anything on this picture so far. I haven't, so this doesn't mean anything to me. But if you've already drawn out roots on your picture, make sure you're clicking the right selection here. So as you can see, the roots from my previous picture have overlaid on this new picture, but they're not correctly positioned. So this root alignment tool will show up here, and you can click on the arrows and move your roots to the proper positioning. So this is a great feature because some of the pictures, when you take the image, they're just slightly off or slightly adjusted differently. So this allows you to move all of those roots at once. So we'll leave that right there. And then we can just exit out of this tool. And now, as you can see, we have quite a bit more roots in this picture. So again, I'm going to click my Add Edit Points tool and just quickly draw out some of these new roots that have popped up in our week 2 image. Here, I am going to zoom in and connect these roots. And I'll quickly just draw out a few more. Again, like I said, if you're doing this with your own images, you're going to want to zoom in quite a bit more and make sure you're adding those points directly onto the middle of the root. And also adjusting those points as well. Okay, so I was able to draw out a few more of the roots. Now we're going to add another session and move on to our final image, our week 3 image, where we can see that there are a lot more roots have developed and are able to be seen in our image. Again, I don't want to have to draw out all the roots that I traced out in week 1 and week 2. So I'm going to right click and again, migrate those roots. With this feature, it's always going to migrate roots from the previous session. So it will migrate the roots only from session 2. Again, I'm going to realign these as best I can and close that out. And then just draw out a few more so that you guys can see the difference in the statistics when we export the data. Another feature about this software is when you start to add some more pictures, it will detect the length of the imager and it will be able to tell you the depth of your root systems. So if I were to add another picture from my tube, we'll just add this one as an example. You'll see that this centimeters up in the top right has changed. So the software knows how long the root imager is. And so for each additional picture that you've taken, it's going to increment the centimeter value here, which will be depth in the soil. When you import your pictures, you can add in your initial window depth here. So maybe you started taking an image with the imager a little bit popped out of the ground, or maybe it was pushed down into the tube a few inches. You can adjust that here so that these depth statistics are accurate when you add new images to your window. You can also adjust the angle that the tube was at. So if it was maybe at a 45-degree angle or something different, you can adjust that here so that these depth values calculate correctly. You can also change the default root status to be, whatever you want here. So I'm going to open up a few of these panels again, just so you can see. With this week 3 image, we've increased our total root area by quite a bit. So if I go back to a previous session, you can see that we have a different root area there. So once we have completed drawing out our root systems and we're ready to export the data, I'm going to just quickly save this to my desktop. And you do need to save before you export. It might take a few minutes depending on how many projects, how many pictures, how many tubes you have in your project. Once you've saved, then you can click File and Export. And it's going to export as a CSV for you. So I'm going to save this. I'm going to stop sharing my screen and I'm going to open up that CSV file to show you exactly what that looks like. So here is my CSV file that I opened that exported to my desktop. You'll be able to see the date and the version up here. And you'll see that you kind of have two different tables. So this top table here is going to be an average of your sessions. So remember, we had different tubes, different sessions for those tubes, and then we had different pictures or windows within that session. So this table up here is going to give you an average for each session that you're in. And it's going to give you things like total root count, total root volume, average density, average length, average width, etc. If we scroll down to this bottom table here, this is where you're going to be able to see the actual statistics for each individual root that you've traced out. So it will have the same naming convention that you saw in the software where the parent roots are a whole number and then any branching roots are incremented with decimals. It's going to give you the length, diameter, area, volume. It'll also give you the root angle and the start and end tip angle. And as we scroll down here, you'll see we have all of the tubes, all of the sessions within those tubes, and all of the windows within those sessions. So you get quite a bit of data from each of those single roots. And any analysis that you need can just be done here because you have all of these statistics for singular roots as well as the entire project. So that was a quick overview and training of our RootSnap software. I'm going to head back over to our presentation here to wrap up and answer some questions about this software. So just to wrap up, I did talk about and introduce our CI 610 autonomous root and soil imager. This has not been released, but we do have links going out to users for people who are looking for more product information or want to be on the list for pricing information and release information. So we have a link for that. We also have a link for product and quote requests with that second link at the bottom. Additionally, I wanted to let you know that we have a link on our website for product consultations. So if you wanted to do a 30-minute call with one of our application scientists. You can book a time through the link on the calendar on our website, and we can have a 30-minute discussion about any of these instruments, any questions that you may have, or just talk about what would be the best instrument for your application. So now I'll go ahead and check the Q&A box for some questions and go through some of those. So the first question we have says, I have a CI 602. I downloaded RootSnap software on your website. However, I cannot use the software to analyze root images. When I open it, it shows that unlicensed copy. How do I download the licensed RootSnap software? Great question. So in a previous release of this software, there was a bug. So when you open the software, there was a little box that popped up that told you that you needed a license key. If that pops up if you have an older version and that pops up, you can just click ignore and continue, and that box will go away and you'll be able to use the software with all of the tools and its full functionality. We did just release a new version that has a little bit of an updated UI and some added tools and features, and that's available for download on our website. If you go to the CI 600 or the CI 602 product page, click product support and downloads, the RootSnap software is available for anyone to download right there. Next from Sam, is it entirely non-destructive? Yes. So when you're using these root imagers, there's two ways that you can install your clear acrylic tubes. The first is that you can pre-install them before you've done any planting. So you have your soil and you use an auger to drill a hole into the soil. You'll place your tube into the soil, and then you'll plant your plants around that tube. And the roots will kind of grow around the tube, and you'll hopefully have a really nice image. Another option is to install the tubes into a pre-existing, garden or orchard farm. And this will be a little bit non-destructive depending on the angle and placing of your tube. But you'll just auger a hole, you'll slide that clear acrylic tube in, and then that becomes stationary. So you don't need to take it out of the soil. It can stay in your pot, in your planter, or out in the field. It has watertight caps on both ends and a foam insert to make sure that no moisture or anything is getting inside that tube. So once you've installed the tube, you don't need to move it at all. And all you have to do is pop off one of the watertight caps, slide the imager right into the tube, and you'll be able to take your images. Next question is, can we measure the height of a tree using this root analysis software? So that is not currently possible with any of these instruments. These instruments are meant to just be essentially a visible image of the roots below ground. You won't be able to get any specs about the actual height of the above ground biomass, as this is purely an imaging tool mostly for soil and below ground root systems. Next question, can it do image analysis of normal JPEG files? Yes. So with this software, you can import any image that you want. It doesn't have to be from one of our root or soil imagers. You can upload any image that you have to this software and start tracing out root systems. Next question, can this tool be used to measure woody tree roots, and what is the depth that it penetrates into the soil compared to the ground penetration radar, which is about 4 to 6 meters in depth? So with these root imagers, as I said, it comes with clear acrylic tubing. And the user has the option to adjust and tell us what size, what length tube they need. So I believe that our standard tube size right now is about 4 feet in length, but you can make that shorter if you need, or we could try to make it longer. The issue with having it be longer than that is that when you're using the imager, it has a metal rod at the top that you use to push down into the tube. And we have multiple that connect to each other, so you always have a grip on the imager. We have made it that length so that you don't lose the imager and you can actually get it back out of the tube. As far as how far outside of the tube you can actually see an image, as the imager is in the tube, it'll illuminate. When you get that picture, you're only going to see roots that are a few centimeters away from the tube. So anything that's right around the tube, you'll be able to see, but nothing farther than a few centimeters away from the actual tube. Next question is, what is the diameter of the area which the system can cover around the scanning tube? Great question. So with our CI600, which is the larger version of our imager, the diameter of the imager and the tube is about 2.5 inches wide. And so that, is going to give you a little bit bigger circumference that you're able to image. With the CI602, it has a bit of a smaller diameter at around 2 inches, and you'll just get a little bit smaller circumference that you're able to measure. Next question, can a machine learning model be written to automatically detect the roots from the soil background without doing the manual tracing of roots? Great question. So we, like I said, are currently in partnership with an AI company to hopefully make this possible. The reason that hasn't already been released or, is not a part of this software already is because it's very tough. With machine learning, you have to add lots of pictures of different root variations, different soil variations, and make sure they're accurately mapped out. So the software can, know what it's supposed to do. And that just takes a ton of data input. With this software right now, users do not have the capability to edit the code or mess with the software. But this feature is coming and we are working on it because we know how time consuming it is to have to sit here and trace out all of these roots. Next question, what if I draw point data for week 3 image and then go to week 1? Is there changes in diameter? How depth soil needs to place the tube? Not sure what the second part of your question exactly is there, but if you draw points for week 3 image, it's not going to adjust anything that you've drawn out on your week 1 image or your week 2 image. So that will stay the same. Whatever you've done on your previous session images will not be changed, will not be edited when you add new sessions, new pictures, new tubes. Anything that you've done on a specific picture will be locked once you move to a new picture. The only option you have for kind of editing those points between sessions is when you migrate roots to a new session. Then that's when you'll be able to edit those points. But like I said, it will not adjust any of the points on previous sessions that you've already drawn out. Next question, I have to measure seminal root angle. Can I use this software to measure seminal root angle? I already have image of roots. Is it necessary that image should be taken from your product? No, so like I said, you can measure, or you can import any image that you have. You do not have to use images from our CI 600 or CI 602. And yes, you can measure root angle. So I only showed you a few times, but to measure root angle, you can draw out the parent root and then use that drag and select tool to drag and drop a branch onto that parent root. And then you'll be able to see statistics about root angle. So only when you start connecting roots to that parent root is when you're going to be able to see that root angle. When you export the data, though, you will get an estimated root angle of all the individual roots. So that feature, you're not able to see that statistic in the software. Only once you export the data are you able to see the angle of single roots. And you'll want to make sure that you have that tube angle feature. You want to make sure you have the correct angle in there as well as the depth so that those statistics are very accurate. Next question, can you save a traced root scan to move it into a PowerPoint presentation? Great question. Currently, we are not able to export the actual traced out image. But kind of a workaround is just using the screenshot tool on your laptop or on Microsoft's, I think it's the snipping tool. So once you've traced out all of your root systems, you can just do a screenshot of your web page, and then that will just, save to your desktop and you can use that for your PowerPoint presentation. Next question, can we also check roots that are very thick and dark? Yes. So the thickness of your roots should not matter. The illumination from the device uses LED. And so we've had really good success with, brown or dark colored roots against dark soil. When those LEDs illuminate, you still can clearly see a difference. And if you're having trouble detecting the difference between, dark roots and dark soil, you can also do some image adjusting before you import it into the software. As I showed you, we have those tools to change the image brightness, the contrast, and the gamma. But if that's not enough, you can, put your image into a different editing software, adjust the colors and the contrast, and then import it back into RootSnap. So you have a very clear image where you can see the contrast between roots and soil. Next question, how is it compatible with UV, Viz, and NIR? So currently our CI 600 and CI 602 are only using visible light. But as I mentioned, we are releasing a new version of this imager, which is our CI 610. And with that version of this imager, you'll be able to use UV, Viz, and NIR wavelengths. So with the CI 610, you'll be able to select specific wavelength ranges that you want, and you'll be able to also select multiple at once. So if you wanted to take a UV, Viz, and NIR image with a single scan, you'll be able to toggle and select all of those on and get an image from all of those wavelengths. But currently right now, UV and NIR are only available on our CI 610, which is going to be released this year. Next question, can I use the root analysis software on 2D hydroponic images? Yes, for sure. So you, like I said, can import any image that you want. If you've taken images with a different device or even with one of our imagers in your hydroponic system, you can import any image. It doesn't have to be roots planted in soil. It can be roots in water or roots in any medium. You should be able to see a very clear contrast between that and be able to trace out all of your root systems. But yes, you can import any picture that you might have from your experiment. Next question, how can the system total area of the root if it will cover a small area around the scanning tube? Good question. So with this technology, the CI 600 and the CI 602 are only able to take visible light images of roots and soil that are directly around the tube. So these tools are not going to give you a complete view of the entire root system. They're only going to give you a view of roots and soil that are directly around the tube. So one way to kind of get around this is just to have multiple tubes. So most, if not all of our users have multiple tubes set up around single plants or all across our orchard. So if you really need to understand what the entire root system of a single plant looks like, I suggest placing multiple tubes around that plant at various angles and positions. So that might be having 6 different tubes scattered around the perimeter of the root system that are all kind of angled in. That might be, adjusting the positioning of the tubes to be right in the middle of the root systems. But having multiple tubes will allow you to analyze all those images and then add all of those statistics together to get a better representation of the entire root system. But right now, we're only able to see roots and soil right around the tube. Next question, is there a way to do image analysis to extract stress-related parameters or indices? Again, another great question. So currently with our CI 600 and CI 602, it's just a visible light image, and the RootSnap software is purely to trace out your root systems and get measurement statistics for those roots. So there's no option to analyze stress or disease or input any sort of indices. But with our CI 610, we're hoping that with UV, Viz, and NIR, we're able to correlate some of those pictures with, different stress, different disease, pigments, etc. On that note, we are looking, like I said, for beta testers who are currently looking at the effects of UV and NIR and how they can correlate those pictures with stress and disease response. So if you are one of those users, researchers, or know of someone who is that would be interested in collaborating with us and testing out this device, please reach out to us. We would love to hear what you're doing and see if you might be a good fit to help us expand the applications for the release of our new imager. Next question, is there any separate lamp replacement cabin or portion? So if there is a issue with the camera or LED lights that are on your device, please reach out to our support team. The link for our support team is on our website, and they will either be able to remotely help you fix an issue with your device or have the device sent in and we'll replace that camera or LED lamp for you. It should be an easy fix. And we always try to help our customers remotely before they have to send in the device. But please reach out to our support team and we'll get back to you if you're having issues. But currently right now, the CI 600 and 602, the lights and the camera are all integrated into one device. They're not separate pieces that the user can take apart. It's all in that one device. Next question is, can I import edited images with lines drawn? Yes. So if you have an image, like I said, you can import any image. So if you're using another software to kind of draw on your image or adjust the settings of the image, you can import any image that you have. You can also, just reopen the project that you have and continue to work on it. So if you only got through tracing half of your image and you didn't have enough time to finish tracing out the other half, you can save and close the project and reopen it and continue to draw out the roots. All the things that you've already done for that project will be saved, and you can just keep adding on to those. But yes, you can import already edited images, images with already roots drawn out. Next question, does that individual root angle give us meaningful biological inference? So that really depends on your research and the application of what you're doing and what you're trying to find out. A lot of users really like that feature because it can tell them different statistics about how, where the roots are leading in terms of direction in the soil, which a lot of times can give users really meaningful information, especially if they have their experiment set up where there's, there's a high nutrient spot in the soil, or they're watering specific parts of the soil. That branching angle and individual root angle can kind of show you where the roots are traveling to and what direction they're pointing. But that really does depend on your application, what kind of statistics you need to prove your conclusion or prove your point in your research. Next question, what's the unique points of your detector over the competitors? So we really pride ourselves in having completely non-destructive tools. We don't want users to have to rip out their entire root systems. Some different ways that people are analyzing their root systems without these instruments are having to completely take out the entire root system from the soil, which you're effectively killing your plant and disrupting the below ground environment. And so we really love that these tools can be completely non-destructive, and also that the clear acrylic tubes can be kind of placed and left, so you don't have to come back and keep digging holes, and it doesn't take a lot of dirty work. You have your clear acrylic tubes that are left out in the field or left in pots or planters that you don't have to touch once you've installed them, which makes it really easy and accessible for you to go out into the field and just pop that imager down in the tube. We also love that these imagers are super portable. Honestly, I just throw it in the cup holder side of my backpack and it fits right in there, and then throw, the laptop or tablet in my bag, and you're good to go. It's really nice that you can just kind of throw it in your backpack and you don't need a whole carrying case or, big kind of box to move it around. Another unique point is that we have this, compatible RootSnap software. So you're able to take these images and then with, a software from the same company, you're able to actually analyze those root systems and, get help with both your instrument and the software. So that's, a few pretty unique points about CID Bio-Science and our line of root and soil imagers. Next question, I already have the tubes in place in the ground to be used with one of your current root imaging models. If I buy the new CI610, can I use it in the same tubes I already have, or do I need to install new tubes of a different diameter? Again, another great question. So with the CI610, the CI610 is going to be the same diameter and is going to use the same tubes as our CI600, which is our wider diameter imager. So if you already have a CI600 and already have tubes installed for that CI600, if you were wanting to purchase the CI610, you would not need to get any more tubes. It will be the same exact size as the ones you already have installed. If you have a CI602, unfortunately, right now, the 610 will not fit in those tubes. But we're hoping that once we release the 610, we can work on releasing another version that's narrower that will be able to fit in those 602, which are those smaller diameter tubes. But right now, only the 600 tubes will work with the 610. Next question, can we connect these multiple images to have the whole area or complete root picture? So within the software, there's no option to actually physically connect the images together, but that's what that Windows option on the left-hand side of the screen was, so that you can import multiple pictures from the entire length of the tube. So let's say you have 3 pictures from a single tube. You can import all of those, first picture, middle picture, then last picture. And they'll show up in a line on that window section. And then once you trace out all of those roots and export the data, it will average all of the statistics for all of those pictures at once. So instead of actually stitching the actual images together, when you export the data, it will just average and give you the statistics for that entire tube. So all the images that you've traced out in that tube. You can also, trace out all of your roots, take a screenshot of that picture, and then take screenshots of the other 2 pictures and use a different software, or just place them right on top of each other so you can have a visual image of your traced out complete root system. Next question, does the angle setting 0, 45 angles in the CI600 affect any parameters of the roots? So the angle setting, when you place your tube in the ground, you're gonna wanna note the angle that it's at and input that into RootSnap. This is really only important if you are interested in the depth statistics or the branching angle statistics. You want to make sure that you have that angle setting accurate so that the, root angle and depth statistics are accurate, but it will not affect any other parameters like diameter, length, etc. You only want to make sure you have that if you're very interested in depth and angle of the roots. Next question, would you anticipate the release of the CI610? So we're not sure exactly when we're going to release this right now. We really want to make sure that this is a well-rounded product that can, has an easy interface and can take images with UV, Viz, and NIR and do it well. Our engineering team and application science team is working really hard to get this device and release it to everyone. But before we do that, we're going to do all of our tests and do beta testing and make sure that it has all of the functionalities that we want it to. Right now, we're hoping, maybe fall 2023, it will be released, but please, sign up for our early bird info so you get all of the information about, when we're going to start beta testing and when the potential release of that will be. Next question, by using this software, can we see the bacterial root colonization? Good question. So with the CI 600 and CI 602, it's really going to depend on the image resolution that you're using. So like I said, the CI 600 only goes up to an image resolution of 600 DPI, but the CI 602 goes up to 1200 DPI. So if you are a user or a researcher who is really interested in mycorrhizal interactions or looking at, specifically at root hairs, I definitely suggest getting the imager with a higher image resolution. You'll be able to zoom in and, kind of see what's going on at a microscopic level. You might not be able to see all bacterial root colonization because it will be very small. But even with 600 DPI, I've had really good success being able to very clearly see root hairs and small differences zooming into the picture. So definitely suggest, taking images with the highest image resolution as possible. And I saw that next question, can we measure root hairs? So like I said, just making sure that you're using the highest image resolution that you have on your device. But as you can see, the background image on the presentation right now, that image was taken using the CI 600 at 600 DPI. And you can see that you can vary, even at this macroscopic view where I'm completely zoomed out, you can kind of start to see some of those hairs on the roots. And then once you zoom in, you can very clearly see these individual roots, and you can even trace them out and map them out if you want to. Next question, do you expect the images from the CI 610 to be different, more compact, etc.? So we're not exactly sure yet. Hopefully they'll be, the same type of image, but it will be, you'll be able to see it from your mobile device, and you'll be able to get an image from a visible light image, a UV image, and an NIR image. We don't know exactly what the size of those images will be yet, or, how they'll be able to be transferred, like what the size, but it should be, just like the CI 600 and CI 602, you're just taking a single image. But with the CI 610, you're going to get a video and a picture. So as it travels down the tube, it has a fisheye lens at the bottom. And so you'll get a video of the entire length of the tube, and then it's going to process those images into individual pictures. Next question, is condensation a possible problem while taking pictures with the CI 610? If so, how should I handle this problem? So this is a possible problem for our whole line of imagers. So the CI 600, the CI 602, and then eventually the 600 or the 610 as well. There's a few ways that you can combat this problem of condensation or soil particles getting inside the tube. So first, when we ship the tubes to you, there is there's two watertight caps on both ends. One of them is removable so that you're able to actually slide the imager into the tube, but it's connected to a foam insert. So that foam insert will take up the entire length of the tube. And we've added that to absorb any condensation that might seep through and get into the tube. We also have a microfiber cloth that we send with you in the box, and a little metal rod that you can thread that cloth through, and then stick it down into the tube and swab along the side of the tube. So if you get any water condensation in there, like I said, you can just use that microfiber cloth or use any soft towel to kind of get in there and get that condensation off the sides. Next question, if we could not have the whole root image, that means we cannot have the area of the root or the total volume, and the customer will destroy the root to get this information and complete his research requirement. Yeah, so like I said before, with these instruments, you're only getting an image of the roots that are directly around the tube. And so to combat this problem, you need to install multiple tubes around your root system to capture as much of the root system as possible. It's not possible to, non-destructively get an image of the entire root system with these devices. But, like I said, you can just install multiple tubes. If you are a user or a researcher that absolutely has to look at the complete root system, then you would need to use a different method, whether that be a destructive method or a different device. Next question, how much is the overlap between two sequential depth images? So this is going to depend on how far the user is pushing the device down into the tube. So like I said, we have these metal rods that come with your imager, and they are the exact length of one tube length. So as you connect more and more, we tell users that, first you'll start the imager at the top of your tube, and then you're going to take that rod and push it down to exactly where the top of the imager was before. And so this is to make sure that you're able to, note the length of your tube and make sure you're getting an image of the entire length. But that overlap between images is really going to depend on how far the user is pushing the imager down the tube and the accuracy, of where they're taking the images. But if you, mess up and you notice some overlap in your images, you can always adjust the size of your images, so crop them and then import them into the software, just like any other image. Next question, is there a possibility to configure or calibrate the measurements if the software is used with your own pictures? So there's no need to configure or calibrate. You might have to adjust the resolution of your picture if you're using an image that's not from our device, but there's no need to calibrate or configure anything. If you're unsure about the measurements, we have that manually measure tool in the software where you can quickly kind of just drag it over the length or width of your root and then draw out your root system and compare those two. But there's no need to calibrate or configure any parameters of the software to make it work with your own pictures. Next question, how would the system work in a cold or frozen environment? Great question. So these devices have a pretty broad range of operating temperature. We have users who are using this device in frozen tundras, in the Arctic. Once you install your clear acrylic tubes, it's kind of just a place and leave type thing. And the tubes will be fine in pretty much any temperature. If you're worried about a specific environment or operating conditions, you can always reach out to us and we can kind of, figure out if this is going to be a good fit for you. But we have users who have been using these devices in a very broad range of temperature, from frozen environments to very hot and humid environments. Next question, can one estimate root biomass from images using the software? So there's a few different statistics in the software that can be used to correlate root biomass. Once you've drawn out all of your roots in your picture and export the data, you're going to get an average value of root density, root mass, and you can also estimate the root percentage of your, how much the roots were taking up in your picture. And you can, correlate that to root biomass. Like I said before, you're not going to be able to get, circumference or anything of these roots because it's just a visible light image. But you're able to get some pretty good statistics about density and mass and even percentage from your picture. So that looks like all of the questions that we have today. Thank you everyone for joining us for our RootSnap live demo. Please look out for information from us on our CI 610 release. And if you want to receive a quote or want to receive information about any of our products, make sure you use this link at the bottom of the presentation. And you can always reach out to our support team via our website or make a time with one of our application scientists, again, with the link on our website. Thanks, everyone. Hope to see you in our next webinar.